general and its own R&D centers in particular. With its headquarters in Thiruvananthapuram, 8 R&D centers, 3 grant and aid institutions, 2 centers of excellence are functional under the Council. KACST is also designated as the special purpose vehicle responsible for executing the 3 science parks in the state. Sophisticated Test and Instrumentation Center at CUSAT Cochin, Community Agro-Biodiversity Center at MS Swaminathan Research Foundation at Wayanad, and Integrated Rural Technology Center at Palakkad are the grant and aid institutions of KACST. KACST has also facilitated the establishment of Sri Chitra Tirunal Institute of Medical Science and Technology, Rajiv Gandhi Center for Biotechnology, National Center for Earth Science Studies, and the Institute of Advanced Virology. At the headquarters, we initiate several programs to advance the science and technology objectives of the state. We have fellowship programs for students and researchers, research funding schemes for mid-career researchers, and programs exclusively for women scientists. To support R&D initiatives from superannuated scientists, we operate the Emeritus Scientist Scheme. Selective Augmentation of Research and Development and Shastra Poshani, science labs for government high schools in Kerala, are 2 of our science infrastructure development programs. Under Sreshta program, KACST nurtures centers of excellence for world-class research in various domains. The Council also supports programs for technology development and innovation and environmental conservation. The Kerala Biotechnology Commission supports biotechnology development programs in the state. Activities of the Patent Information Cell and Intellectual Property Rights Information Center Kerala facilitate IP awareness and patent-related activities. KACST Scheduled Caste and Scheduled Tribe Cell is a DST-supported project that provides S&T solutions for the development of SC/ST communities. Science popularization activities like KCST Shastra Puraskaram, Kerala State Young Scientist Award, and Science Literature Awards are notable activities of the Council. KCST is honoured to recognise the legendary scientists and science advocates through the Kerala Shastra Puraskaram. The National Children's Science Congress, Technology Festival, and Rural Innovators Meet are other major science popularization programs. KACST also supports the observation of National Mathematics Day and National Science Day programs in the state. Kerala Science Park is a new initiative facilitated by KACST for R&D development in the state. The activities of 3 science parks, Bioprocessing and Bioprospecting at Kannur University, Materials and Marine Sciences at Cochin University of Science and Technology, Green Energy, Healthcare, Genomics and Planetary Sciences at Kerala University are ongoing. The Center of Excellence in Microbiome and the Center of Excellence in Nutraceuticals are the other new initiatives of the Council. The Kerala Science Congress is an annual event conducted by KACST that provides a platform for young researchers, academicians, technologists, and others across the state and elsewhere to discuss, exchange, and share their research findings and technologies developed relevant to the state of Kerala. Last year, the 36th edition of Kerala Science Congress evinced the presence of 2022 Nobel laureate in chemistry, Professor Morton P. Meldal, addressing the gathering. The 37th edition of Kerala Science Congress is scheduled from February 7 to 10, 2025, with the focal theme Technology Transformation for a Green Future. Kerala State Council for Science, Technology and Environment, advocating science and technology innovation in the service of the state of Kerala.
May I invite the dignitaries, Professor Joby K. Jose, Professor A. Sabu, Professor Anup Kumar Keshavan, Professor P. Balaram, Dr. P. Harinarayanan, and Dr. Suraj M. Bashir to occupy the dais.
May I invite Dr. Suraj M. Bashir, Associate Professor and Head, Department of Molecular Biology, Kannur University, for the welcome address. Welcome, sir.
Okay. Dignitaries on and off the dais, teachers and students from our university and various affiliated colleges, and non-teaching staffs. Good afternoon, everyone. A hearty welcome to one and all to the G.N. Ramachandran Memorial Lecture organized by Kerala State Council for Science, Technology and Environment in association with Kannur University. This memorial lecture is a curtain raiser for the 37th Kerala Science Congress to be held in the month of February at Thrissur. Today, we are gathered here to celebrate the legacy of Dr. G.N. Ramachandran, one of India's most revered scientists, whose groundbreaking contributions to molecular biology and structural biology, including the iconic Ramachandran plot and the triple helical model of collagen, have left an indelible mark on science. This memorial lecture pays tribute to his remarkable work and serves as an inspiration for future generation of scientists. Let me come to my duty of welcoming each one of you to this function. The president of today's function is our registrar, Professor Joby K. Jose. He is a professor of statistics and one among us, and he is very supportive in all our endeavors. On behalf of KSCST and on my personal behalf, I extend a hearty welcome to Professor Joby K. Jose to this program. We are truly privileged to have with us today Professor P. Balaram, an eminent biochemist and molecular biologist, and a stalwart in the field of protein chemistry and bioorganic mechanisms. Professor Balaram's extensive body of work spanning decades has not only deepened our understanding of protein folding and interactions, but also positioned him as a leading figure in global science. It is indeed an honor to have him deliver the G.N. Ramachandran Memorial Lecture here. On behalf of Kannur University and KSCST, Government of Kerala, I extend a hearty welcome to Professor P. Balaram. We have Professor Sabu with us, a former Pro-Vice Chancellor, former IISc Director. He is the Professor of Department of Biotechnology and Microbiology and currently on deputation as Member Secretary, Kerala State Council for Science, Technology and Environment, Government of Kerala. We all know the efforts taken by him which helped to improve our NAC and NIRF rankings. He is the General Secretary of 37th Kerala Science Congress. And on behalf of Kannur University and KSCST, I extend a hearty welcome to Professor Sabu to this program. Also, I take this opportunity to thank KSCST for providing an opportunity to Kannur University for organizing this significant event as part of the Kerala Science Congress. Professor Anup Keshavan is the Director, Internal Quality Assurance Cell of our university. He is a professor in the Department of Biotechnology and Microbiology and was a former Director of Research Directorate. A hearty welcome to Professor Anup Kumar Keshavan to this program. Teachers and students from all the science departments of our university have gathered here today. Not only from our departments, many teachers and students from our affiliated colleges are also here. Dr. Harinarayanan, Senior Principal Scientist, KSCST, is here with us. A hearty welcome to Dr. Harinarayanan to this program. Sorry. Yeah, so I was telling there are students and teachers from our university and from affiliated colleges also. And there is representation from Central University of Kerala. We have Senate syndicate members here. On behalf of Kannur University and KCST, I extend a hearty welcome to all the teachers and all the dignitaries to this program. So once again, a hearty welcome to one and all to this program. Thank you.
Hello. Thank you, Dr. Suraj M. Bashir, for your warm and gracious welcome. Next, we are happy to invite Professor Dr. Joby K. Jose, Registrar of Kannur University, to deliver the presidential address. Please, sir.
Today's chief guest, Professor P. Balaram, Dr. Professor A. Sabu, Member Secretary, Kerala State Higher Education, Kerala State Science, Technology and Environment, Sri Harinarayanan, Senior Principal Scientist, KCST, Sri Suraj M. Bashir, Head of the Department of Molecular Biology, Sri Anup Kumar Keshavan, Director of IISc, and Anish, the member syndicate, and deans, professors and faculty members from various departments and other colleges, and dear students. As all of you know that the 37th edition of Kerala Science Congress is going to happen in February 7th, 8th, 9th and 10th at Agriculture University, Thrissur. And Kerala Kerala Council for KCST is actually organizing the Kerala Science Congress. KCST is a forum in Kerala which is mainly for popularizing the science and the research activities of Kerala actually. In connection with the Science Congress, KCST is organizing the memorial lectures. I think actually it is 9 memorial lectures they are organizing, and it is the third one in this edition of the Science Congress. The first one they organized at the Kerala University, and it was on Janaki Ammal endowment lecture. The second one at Calicut University, P.R. Pisharadi endowment lecture, and the third one is on Professor G.N. Ramachandran endowment lecture. Actually, today we are very privileged because for delivering Dr. G.N. Ramachandran endowment lecture, the former director of the Indian Institute of Science and the very famous scientist of India, Professor Balaram sir, is with us. First of all, I take this opportunity to thank Professor Balaram sir for arriving here and agreed to deliver a talk in the forum in this August, in front of this August audience of Kannur University. Thank you, sir. I know that this is not the time for an elaborate speech. You are all eagerly waiting for the words of Professor Balaram sir. And one thing is, I remember that this is a forum, this is a venue, it gives us opportunity to discuss with professors like Professor Balaram and how they approach the research problems, what are their contributions, and how the science developed in India and all over the world. With these small words, I invite Professor Balaram sir once again and wish you all successes for this program. Thank you very much.
Thank you, Professor Joby K. Jose, for your insightful and motivating address.
To remember the legacy of Dr. G.N. Ramachandran, an eminent physicist whose contributions revolutionized structural biology, We now invite Professor A. Sabu, Member Secretary, KSCSTE, and General Convener of the 37th Kerala Science Congress to give an introduction to the life and work of Dr. G. N. Ramachandran. Please, sir.
Good afternoon, most respected Professor P. Balaram, Professor Joby K. Jose, Registrar, Kannur University, my colleagues and good friends, Professor Suraj Bashir, Professor Anup Kesavan, and my colleague at KSCST headquarters, Dr. Harinarayanan. Let me thank Professor Joby and his team for coming forward to host the memorial lecture, which is part of the 37th Kerala Science Congress scheduled to be held at Kerala Agriculture University campus from February 7th to 10th. This year's Science Congress activities were kicked off on 6th of December by organizing the Dr. Janaki Ammal Hello. Sorry for the interruption. So this year's Science Congress activities were kicked off at the University of Kerala by organizing the Dr. Janaki Ammal Memorial Oration. The lecture was given by Dr. Jawahar Ali, a reputed plant breeder from the International Rice Research Institute in Philippines. The second memorial lecture was given by Dr. Tamban Melath, Director, National Center for Polar and Ocean Research, Ministry of Earth Sciences. The third memorial lecture, we are fortunate to have Professor P. Balaram here. Thank you, Balaram sir, for accepting our invitation to visit our university and to give this lecture on behalf of KSCST. Let me take the proud privilege to introduce Professor G. N. Ramachandran. Professor G. N. Ramachandran was one of the most intellectually gifted scientists of independent India. Born on the 8th of October 1922, he has completed his schooling from Esvale Princely State of Cochin. He then completed a BSc Honours degree in Physics from St. Joseph College, Trichy, in first rank. In 1942, G. N. Ramachandran enrolled for a Master's degree in Physics at the Indian Institute of Science under the guidance of Professor C. V. Raman. His Master's thesis titled Optics of Heterogeneous Media was received with great accolades. He continued his research at the Institute for a doctoral degree. G. N. Ramachandran followed this with a second doctoral degree from Cambridge University in the year 1949. At the early age of 27, G. N. Ramachandran was a double doctorate in X-ray, specializing in X-ray diffraction studies. After a brief stint as an assistant professor in the Department of Physics, Indian Institute of Science, Professor Ramachandran moved to the University of Madras as professor and head of the Department of Physics in October 1952. Professor Ramachandran, along with his peerless postdoctoral fellow, Dr. Gobinath Karta, determined the three-dimensional structure of the structural protein collagen, which is arguably the first structure of a protein ever to be determined. This was followed by several studies on the structure and conformation of proteins and peptides, which culminated in the formulation of the eponymous Ramachandran plot that is now known to every student and practitioner of biochemistry and protein chemistry. In 1971, Professor Ramachandran returned to the Indian Institute of Science as the founding head of the Molecular Biophysics Unit. He nurtured strong groups of faculty members in experimental X-ray crystallography, protein chemistry, and biomolecular confirmation using computational methods. Since then, the molecular biophysics unit has grown to be one of the important centers for structural biology in our country. Towards the later stages of his career, Professor Ramachandran pursued his love for mathematical philosophy and continued to work independently on various problems in logic and philosophy. Professor G.N. Ramachandran is the recipient of several scientific awards, both national and international, such as the Shanti Swarup Bhatnagar Prize, the Meghnath Saha Medal of the Asiatic Society, the Srinivasa Ramanujan Medal of the Indian National Science Academy, the Sir C.V. Raman Medal of the Indian National Science Academy, and the Fagarty International Medal of the National Institute of Health, USA. I'm not narrating all the awards won by Professor Ramachandran. He is also an elected Fellow of the Royal Society in the United Kingdom. Friends, I think it was in 1991 when I joined for my master's in biotechnology at Cochin University of Science and Technology. I started learning about Professor G.N. Ramachandran and the Ramachandran plot. That time, it was Professor Joseph D. Padayati from the Indian Institute of Science who taught us biochemistry. And this is a great occasion. After 34 years, I am standing here in front of you to organize a memorial lecture in the name of Professor G.N. Ramachandran. And I am so happy and proud that another eminent scientist of the country is giving the memorial lecture. Thank you very much.
Thank you, Professor A. Sabu, for the inspiring words and reminding us the remarkable contributions of Dr. G.N. Ramachandran.
It is our pleasure to invite Professor Anup Kumar Keshavan, Director, IISc, Kannur University, to introduce today's distinguished speaker, Professor P. Balaram. We invite you, sir, to introduce our renowned speaker.
Respected dignitaries on the dais and off the dais, Registrar Joby sir, Chief Guest Balaram sir, Member Secretary Sabu sir, Dr. Suraj, Dr. Harinaran, and off the dais, Syndicate Member Anish Kumar, HOD and faculty members of our department and from the other colleges as well as other universities, my dear students. Actually, it's a privilege to introduce Balaram sir to you, because in the morning I had the same privilege to introduce him to all the faculty members of ours. So without wasting much time, let me go in a nutshell. What has Balaram sir done throughout his life till now? Professor P. Balaram is a distinguished biochemist and molecular biologist and a former director of Indian Institute of Science, Bengaluru. He made significant contribution in the field of protein chemistry, molecular biophysics, and bioorganic chemistry. A prolific researcher, he has authored more than 600 research papers and is a fellow of prestigious scientific academics, including the Indian Academy of Sciences, Indian National Science Academy, the World Academy of Sciences, Trieste, Italy. He is also the editor of the journal called Current Science for the last 18 years. His exceptional work has been recognized with numerous awards and honors, including Shanti Swarup Bhatnagar Prize, the TWAS Award in Chemistry, Padma Shri and Padma Bhushan from the Government of India. He has been conferred the 2021 Bruce Murphy Award by the American Peptide Society. Professor Balaram has served on various national and international committees, including the Scientific Advisory Committee to the Union Cabinet and the Scientific Advisory Council to the Prime Minister. He is currently serving as a visiting professor at the National Center for Biological Sciences, Bengaluru. Once again, we welcome you to Kannur University for delivering this lecture, sir. Thank you so much for accepting it.
Thank you, Professor Anup Kumar Keshavan, for the detailed introduction.
We now come to the highlight of today's event, the G.N. Ramachandran Memorial Lecture. May I invite Professor Balaram to deliver the memorial lecture.
I'll use that. Can I just use this?
Oh, yeah, fine.
I'll use this. Because that I don't like.
Oh, yeah. Slide.
Aye. Okay, good afternoon. I'd like to thank the Kerala State Council for Science, Technology and Environment for inviting me to give the G.N. Ramachandran Memorial Lecture as part of the Kerala Science Congress. My special thanks, of course, goes to Professor Sabu for inviting me to this event. And for someone like me who was quite old, coming here to speak about G.N. Ramachandran actually takes me back to the beginning of my own scientific career, because Professor Ramachandran hired me as a lecturer in his newly started department, the Molecular Biophysics Unit at the Indian Institute of Science. And so after all these years, I am standing before you to tell you about what Professor Ramachandran did before he came back to Bangalore and set up our department. And much of the work that he did really was done in Madras. Kerala has claimed G.N. Ramachandran as a distinguished son of Kerala, and indeed he is, because he was born here and he had all his early education in Ernakulam. But at the same time, it turns out that he went to college in Tamil Nadu, and all his famous work was done in Chennai at Madras University. Tamil Nadu has not yet claimed him, but at the Indian Institute of Science, of which I was the director many years ago, we always claimed G.N. Ramachandran as one of our alumni. And he was in fact the first recipient of the Distinguished Alumnus Award instituted by IISc many, many years ago, because that's where he really came into science and did his PhD, working under the guidance of C.V. Raman. Now, what the first slide shows you is really the picture of G.N. Ramachandran in the middle. This is how I saw him when I first met him in 1973. On either side of his picture are the two iconic images which summarize his contributions to structural biology. On the left is the structure of collagen, which I will tell you a little bit about, and on the right is the famous Ramachandran plot. If you don't know anything about protein structures or protein conformations, these are just images. But these are the images which have transformed the field of protein structure over the years. But at the bottom, I have two images again. Those images are small, they are indistinct, and they are handwritten. Those are G.N. Ramachandran's notes in the classes that he used to take when I first joined the Indian. But then I found that he was giving assignments, and he didn't like anybody who didn't do the assignments to come to the class. And I didn't want to do the assignments because I didn't know much about the work that he had done at that time. I later learned about the work that he'd done and used his work in my own research work, which I did over the next few decades. But his work was primarily in the area of protein structures. And I'm going to show you only one slide which is not concerned with G.N. Ramachandran to illustrate the importance of proteins. And there is no better example than to take the example of the COVID-19 pandemic. Because in the COVID-19 pandemic was driven by the coronavirus. And the coronavirus infected all of us. I don't believe there is anybody in this audience who was not at one time or the other infected by the coronavirus, even if they did not fall sick. I've called this slide Viral Host Fusion and Wonderland, because when you read about this, it really reminds you of Alice's Adventures in Wonderland, Lewis Carroll's famous book. The virus is pictured there, and the virus is best defined by something that Peter Medawar, the English immunologist, wrote many years ago. He said that a virus is a piece of bad news wrapped up in protein, which means this virus has its genetic material circular, spherical in size, but the material inside, but the outside of the virus is all protein. Those are those spiky things that you see in red in that famous image which we all saw through the pandemic. It is this protein which is on the surface which allows the virus to seek out the host cell, infect the host cell, and get the nucleic acid inside. And subsequently, there is the production of new viruses. So for the virus to reproduce itself, it has to infect. And in order for it to infect, it needs this protein. But it turns out that the protein is a very interesting protein, because right here, you will see this protein here. There it is. My eyesight's getting poorer and poorer, and I can't see the red dot which is there. This protein looks like this in the virus. Once it's going to infect you and it has bound to the target cell, this is how it looks. So it is a huge elongation of structure, a dramatic conformational change in which the virus is transformed. It's almost like in Alice in Wonderland. Alice will drink something from a bottle and she will become very small. She will drink something from another bottle and then she will become very big. And those are the two images that you see. When Alice sees this phenomenon, she says this, "Curioser and curioser," cried Alice. She was so much surprised that for the moment she quite forgot how to speak good English. And the whole thing's very curious. She said, "I must be shutting up like a telescope." And so today, for scientists who are interested in viral fusion and in understanding enveloped membranes, enveloped viruses, infections in humans, they need to study this protein, and they need to study such examples of conformational changes. Influenza, HIV, the coronavirus infections, all of them are examples of such dramatic conformational changes. The protein size changes in length from a value of 88 angstrom there to 215 angstrom, which is a huge change. So proteins are important. This slide has nothing to do with G.N. Ramachandran, but this slide is to tell you that this protein has driven the COVID-19 pandemic. You might actually call this protein as the protein which is responsible for at least 7 million deaths worldwide. But what did we know about proteins? In 1936, when Ramachandran had just finished school and was entering college, this is what was known about proteins. On the left is a famous book called The Specificity of Serological Reactions, written by the American immunologist Karl Landsteiner. Every one of you knows about blood groups. You know your blood group because it is typed. And you must remember your blood group because in case you need a blood transfusion, it's best if they give you matching blood. Landsteiner discovered the blood groups. When Landsteiner discovered the blood groups and began to think about antibody specificity, the famous chemist Linus Pauling began to think about protein structures, and he collaborated with Landsteiner. And then Landsteiner gave him a copy of his book. And in the Pauling archives, you will find Pauling's copy inscribed with that annotation which you see on the left. What is written over here in hand, that's in Pauling's handwriting. And what Pauling wrote was, I think the protein must contain side chains with active groups. That means in 1936, nothing really was known about protein structures. And over the next 3 to 4 decades, our knowledge of proteins would be dramatically transformed. A little bit of history. In 1943, these were the kinds of pictures that people were writing for polypeptide chains. The hydrogen bond had been recognized as an important interaction stabilizing the structures of molecules. And Morris sort of wrapping around an axis. But the precise nature of that polymeric chain was not quite known. And here is another paper. This is a picture which appeared now in 1950. This is about the time that Ramachandran was going to set out to start his own research work outside the Indian Institute of Science, and the field that he would choose would be to study the structure of collagen. People were studying the structures of proteins, and here's a paper which appeared in the Proceedings of the Royal Society. Why am I showing you this paper? Because this was a very influential paper, and this is the only paper that I know in the literature which has a unique distinction. Every structure printed in this paper is wrong. But even more importantly, of course, for discovering Bragg's law, n lambda is equal to 2d sine theta, which is the basis of crystallography. John Kendrew and Max Perutz for determining the structures of myoglobin and hemoglobin, the first protein structures which were determined in the late 1950s. But in 1950, this is what was known. This is Pauling in Madras now in 1967, because in 1951, Pauling published a paper outlining the structure of the alpha helix. The model that he's holding in his hand was constructed in the workshop of the University of Madras, and Pauling is demonstrating his own structure, 1951 structure, in 1967 when he came for a visit. But the moment the alpha helix came, the field simply exploded after that. It's around this time that Ramachandran, who had done his PhD with Raman, his work was primarily in physics, excellent physical insights, and very good mathematical skills. And it is with this that he went to the University of Madras. But why did he go to the University of Madras? This is because A. Lakshman Swami Madaliyar, the sort of remarkable Vice Chancellor of the University of Madras at that time, had taken over. And he decided that we must start research at Madras University, and the university does not even have a physics department. And physics was such an important subject. So he decided to start the physics department, and he wrote a letter to C.V. Raman asking him to come to Madras and begin the physics department, open the physics department. Raman wrote back saying that he's reached the age of retirement and he no longer can go anywhere. Instead, he recommended Ramachandran. And this is the letter written on the 19th of February in 1952, Raman to Mudaliar, recommending Raman as the professor of physics at Madras University. This you can see is 19th February. A few months later, Raman got the offer. He joined. He was a few days short of the age of 30 when he became the professor and head of the Department of Physics at the University of Madras. Today I know there will be some heads of department here. And heads of department are usually important people. And it will turn out that they control everything in the department. Here you can see there is no department. The department's just going to start. And here you have a young man at the age of 30, now recommended by Raman to start the department. So he's going to come there and start physics, he's going to start research. Because, as I said in the morning, although the University of Madras was set up in 1857, it was set up as a British initiative following the famous Macaulay Minute. There was no research, and research had to be started. And this research started in independent India. And it turns out that Ramachandran's work subsequently at Madras may be one of the brightest events in Indian post-science in the post-independence era. On the right, this is the year 1964. This is the recommendation for Ramachandran in 1952. On the right, in the year 1964, Raman nominated Ramachandran for the Nobel Prize. So you can see that in this very short span of time, of 12 years, Ramachandran had already reached the stage where Raman, who thought that he should be professor of physics, now also thought that he should receive a Nobel Prize for this work. What is it that he did? This is exactly what I will spend the rest of my presentation trying to show you. Pauling's alpha helix, as imagined by Ramachandran, is the sketch on the left, and the collagen triple helix that you see is as he sketched it, thinking about what three chains would be. And I'm going to show you that in a moment. What are the origins of a research problem? How does one start doing research? You have nothing to do. You have no facilities, you have nobody. You must find a problem. And Ramachandran says that he was encouraged to enter this field by reading the papers. So this is the most important thing, read the literature. And you read the literature and find your problem from the literature, not from someone else. Find the problem from your surroundings, but not from someone else. And he chose X-ray diffraction and crystallography because he felt that that was in the early days. He knew, because he'd been in Cambridge, that crystallography was going to be important, because by that time, Max Perutz and John Kendrew had already started their work on the diffraction studies of proteins. I must tell you that the first X-ray diffraction pictures of proteins were taken in 1935 by Dorothy Hodgkin working under J.D. Bernal. But it took another 23 years before the first structure of a protein came. So sometimes major advances in science take a very long time to happen, and there's a large amount of extraordinarily hard work to be done in between. And then he says, however, I did not know where to begin and which molecules of biopolymers were the most profitable ones to study. And he says the problem was resolved by the happy coincidence of a visit of Professor J.D. Bernal to Madras in the early 1950s. So he asked Bernal, what do you think is an unsolved problem? And Bernal told him that collagen structures are unsatisfactory. And then, of course, Ramachandran looked at an environment. Those of you who have been to Chennai, now you will see that it is the old AC College of Technology, which is now Anna University. But in the campus just next, also a fledgling institute in those days. Leather, what is leather? Leather is collagen. And therefore, samples are available. And it turned out that one of the scientists at the Central Leather Research Institute told him, we can certainly get collagen from rat tail tendon. Go back and look at a rat and you will find a tail. You will find material properties of the tail. It's all collagen. It also turns out that Ramachandran also got a sample of kangaroo tail collagen because his friend Dr. Nayudamma, who then became the director of the Central Leather Research Institute, went to Australia and brought back a sample of kangaroo collagen. This then is the collagen triple helix. The only evidence for the collagen triple helix is that fiber diffraction pattern there. It's a long fiber, it has periodicity along the fiber axis, and so it will diffract X-rays. And from those spots that you see, you can in fact deduce with model building the triple helical structure. This is a major achievement at that time. But I want to pause here for a moment because there are two pictures on this slide. There is Ramachandran and there is Gopinath Karta. Now, for some reason or the other, Kerala has not claimed another son of Kerala. Gopinath Karta himself was a remarkable man. He became Ramachandran's PhD student at the Indian Institute of Science. And I'm going to show you a little bit about Gopinath Karta. But before that, a word about collagen. Today, if you look on the internet, you will find these things. So all your tissues made up of collagen. The most abundant protein in animals is collagen. And it's there in bone, tendon, cartilage, teeth, everywhere. It has an unusual amino acid composition. And the only thing you need to know about proteins is that they are polymers made up of amino acids, and there are 20 amino acids which human DNA now codes for. But it turns out largely only 3 of them are used in collagen. Actually only 2, with one of them being modified later on. This is what physics in Madras looked like at that time, and I show this to you. There's a picture taken in 1955. By this time, the structure of collagen had really been solved. You can see Ramachandran sitting in the first row. He's the only person with a tie. Towards the end of my talk, I will show you that as photographs and years have progressed, color has entered photographs. But in India in those days, even when I was young, most of us wore only white clothes because there were no colored clothes and everybody wore white. Some had clean white, others had slightly dirty white, but that was it. And here you are. Everybody is dressed alike. But you can see Gopinath Karta there, sort of proudly establishing that he is from Kerala. And he has, in fact, he's dressed appropriately and he's a key figure in the structure of collagen. There are others whom I've indicated. During the pandemic, when I had a fair amount of time, I had the opportunity to correspond with Dr. Karta's wife in America and Karta's former colleagues in America, and wrote an article about the roots of chemical crystallography in India, a historical note on Gopinath Karta. I show you his molecules in a moment. But that's the triple helix auditorium in Chennai. Of attending the inaugural of this auditorium as a substitute for Ramachandran, who was by that time too ill to be able to travel to Chennai for the inauguration itself. But you can see that this is remarkable because it's the only auditorium that I know in India which is not named after a person but after a molecule. There can be no greater tribute to science than this. This slide is what I would call Karta's molecules. Karta began his career under the mentorship of G.N. Ramachandran. He determined the first crystal structure in India. This is the structure of barium chlorate monohydrate, and that's his PhD thesis. It's a PhD thesis from the Indian Institute of Science, but in those days the Indian Institute of Science did not have the legal authority to give degrees, so the degrees were given by the Madras University. Instead of staying at the institute or going abroad after his PhD, he followed Ramachandran to Madras as a postdoctoral fellow with his PhD. And he did collagen. Ramachandran and Karta also did the first natural product which had been determined in India. That is a natural product called morellin, which is from the plant Garcinia morella. This is remarkable because chemical crystallography was difficult in those days, and this was a difficult molecule, and it's the first member of a family of molecules called the caged xanthones, which are important today. And then when he went to America, he solved the structure of ribonuclease. It turns out that ribonuclease is the first protein structure solved in the United States. So all the structures till then had been solved only in Cambridge, England, and Karta did the enzyme first structure of ribonuclease. So that's ribonuclease. So when you look at this, you immediately get the feeling that Ramachandran and Karta were two men who came together, very young age, but both of whom had remarkable scientific abilities and incredible insights into the way molecules are constructed. But coming back to collagen, the peculiar amino acid composition of collagen is that it has glycine, proline, and hydroxyproline repeated. Acids which stand out are glycine and proline. Glycine because it's very small, and proline because it does not have an NH group. The result of this is any structure formed by collagen must be a unique structure. The amino acid composition is unique in that 1/3 of the amino acids are glycine. That means 1/3 of the amino acids must be very small. What does this tell you? If you have a threefold structure, that means the same thing must be repeated 3 times. So instead of 1 helix, you can begin to think about 3 helices with the smallest amino acid put in the middle, where there is place to accommodate it. You can't put a bigger amino acid. And these were the ideas which Ramachandran and Karta used to publish their first paper in 1954 in Nature on the structure of collagen. They had determined the unit cell dimensions. Unit cell dimensions means a unit cell is like a box. And this particular box, because of the symmetry and the space group, had only 2 axes. 2 of the axes had the same value. So it was a box which was 2 of the axes had 12 to 16 angstrom. The third axis was 9.5 angstrom. So now you know the volume of the box. And once you know the volume of the box, you know what can fit inside that box. And this must repeat. And what would fit inside that box was only the tripeptide, that is glycine, proline, and proline. So you must have an infinite repeat of glycine, proline, and hydroxyproline in this in the collagen structure. And their mathematical abilities, they were immediately able to build in projection a structure like this. You can see the threefold symmetry in some of these structures. Now there are a series of papers here in the period between 1954 and 1956 from Madras and Nature, in which the collagen structure was outlined. Collagen now is the triple helix of collagen. But there's a twist to the story. There are 2 more papers at the bottom here. Those 2 papers are by Francis Crick and his colleague Alexander Rich. Alexander Rich became a famous person in later years, but Francis Crick already in 1955 was the man who, along with Jim Watson, had produced the DNA double helix. So Pauling had produced the alpha helix in 1951. Watson and Crick produced the DNA double helix in 1953. Ramachandran published the triple helical structure of collagen in 1954. So you can see these are all contemporary structures at that time. But what Crick and Rich working on collagen said Chandran structure, and that is because in that structure, the atoms come too close to one another. There's no place for atoms. You know, when you think about atoms, just think about atoms like you think about tennis balls or cricket balls. You can't compress 2 cricket balls too much. 2 tennis balls you might be able to compress just a little bit, but 2 balls cannot all contacts like this. That's the wire model of collagen, which I already showed you. And then Ramachandran sent his paper to Pauling, and Pauling also said, maybe the structure is not completely satisfactory. I think at that time, people had not thought about the triple helix properly, and they felt that maybe there were still some problems. But here is a letter that I show you, and this letter came in 1955. 1955, Francis Crick at Cambridge organized a seminar on the structure of collagen, and he invited Ramachandran to attend. And he said, we should be very glad to have your comments on these remarks. We are holding a seminar here, and we would like to present your latest ideas. At this time, Karta was visiting Cambridge. And he said, or maybe you would prefer that Dr. Karta present the results. This is when the now, of course, when you look at this letter, it's marvelous. I'm sure all those of you who are younger never have seen a letter like this. And when we all used to write letters, we used to use the space effectively because an aerogram cost money. So you would put as much information as possible into whatever letter you were writing. And Karta wrote to Ramachandran in 1955, but I've only taken out what could be deciphered easily. He says, Dr. FHCC, that's Francis Crick in Cambridge, was telling me, and he says, I do not know what he meant, but the general idea seems to be that publication of our paper was purposely delayed by months for them, Randall at King's College London, to work up our idea after seeing our manuscript. And this is how things used to happen then. Randall is the same person in whose laboratory Rosalind Franklin took the famous DNA photographs, which eventually became photograph 51. Groups, I would prefer a 2-hydrogen bonded system, etc., etc. That's technical. So Karta and Ramachandran were now in correspondence with one another, while Crick and Rich were now challenging the structure. But eventually the Ramachandran structure turned out to be right. But you can imagine the effect that this would have, because in India, of course, the word of the Westerner is always fine. Us were rather happy that a young man, an upstart professor and head of Department of Physics, has actually stumbled on something that appears to be major. And there would be a lot of murmuring, and this, I think, affected him quite substantially. But many years later, he said that everything's become very interesting, exciting at this time. The double helix was proposed, and the helix era had begun. And so what I will show you on the next slide are the 3 helices, which are now immortal in biochemistry textbooks. The first is the Pauling alpha helix in 1951, then the Watson-Crick double helix for DNA in 1953, and the Ramachandran triple helix for collagen in 1954. One cannot but feel a bit of pride when one sees this chronology. Linus Pauling, many years later, said that in 1951, he and Professor Corey had proposed a structure which involved 3 polypeptide chain. It is well known that our structure turned out to be not right. And then he said that they should be given more chances. But he says, before that, Ramachandran had already done the structure. He says, although I may have some feeling of regret that we did not succeed in making our second guess, which I trust would have turned out to be the right one, great credit must be given to Ramachandran. This is what he said when he came to Madras in 1967 for an international conference. But here I must tell you that there were 2 conferences held at Madras, one in 1965 and the other in 1967, which brought almost every major figure across the world to Madras to attend the international conference. And you can imagine those were not the days in which 5-star hotels were easily available, flights were not easily available, and for the world scientists to make the trip to Madras would have been only because a major work had been done then. Francis Crick, in his final autobiography, wrote this. He says, what was important was not the way it was discovered, but the object discovered, the structure of DNA. And then he says, consider the discovery of the basic structure of collagen. And then he says, the characters were just as colorful and diverse. The facts were just as confused and false solutions as misleading. Competition and friendliness also played a part, yet not a single book has been written about the race for the study of collagen. Whereas Watson wrote the book, The Double Helix, and molecular biology really began with that book. And then he says it depends on what you're doing. He says in 1955, after we got interested in collagen, we began saying, do you realize that 1/3 of the protein in your body is collagen? Before that, they said there is no collagen in plants, and so it's not important in biology. DNA, of course, is present everywhere. This tells you a little bit about how science works. And he says it is the molecule that has the glamour, not the scientists. And at least I think we need to invest collagen with a little bit of glamour, because it was in fact truly an Indian discovery. Many years later, in 2012, long after Ramachandran had passed away, this structure appeared, which is a single crystal of a fairly long collagen stretch, which established that the Ramachandran structure was essentially correct. So he says here, for example, this is what he says. That's what it looks like. But the controversy on the collagen structure led to this idea that if you were going to evaluate the structures of proteins, how do you know that they're right? And the question really asked was, how close can two atoms come to one another? And you will of course come to the conclusion that two atoms cannot come closer than the sum of their van der Waals radii. Now I do not know how many of you, because many of the students here may be students of biology, and if you are students of biology, sometimes you might take atoms for granted. I might actually ask you a question, are atoms important? You might say, well, yes, they are. But in order to emphasize the importance of atoms, I will give you a reference. And this is to the Feynman Lectures in Physics, Volume 1, Chapter 1. Feynman begins his famous lectures on physics with a statement, with a question. The question he asks is this. He asks that if in some cataclysm, all of scientific knowledge were to be destroyed, what is in one sentence, what is the information that can be conveyed to the next generation so the entire edifice of science is built up once again? It's an interesting question, because Feynman was worried about the Cold War between Russia and the United States, Soviet Union and the United States at that time, which could have resulted in a nuclear conflict. This was shortly after the Cuban Missile Crisis of 1962. We should still be worried about it because we have wars raging everywhere and everybody's got a nuclear weapon. So this could happen. He then answers his question. He says, if all of scientific knowledge were to be destroyed and you wanted to start all over again, we need a clue so that we can go faster. And he says, I believe that clue is the atomic hypothesis, that all things are made up of atoms, and that's the most important thing. With that one statement that all things are made up, and so the van der Waals contact distance is about the closest that you can squeeze two atoms together. So Ramachandran asked this question, what is the closest that you can bring two atoms together? But then at that time, nobody knew what the van der Waals radius of an atom was, or there were numbers. Bundy's numbers were there, but they were not satisfactory for looking at biological structures. So his students and he went back to the literature. There was very little crystallographic information. And they asked the question, how close do atoms come in crystal structures? Because you know, in crystals, molecules are packed into one another. And non-bonded atoms must come within the van der Waals distance. It's very, very important to understand that one thing which you must remember is nature does not like vacuum. In nature, everything sort of packed reasonably well, and you don't have empty spaces everywhere. And this is true of crystals. And so they got experimental van der Waals contact distances from crystal data, and then made models of peptides and came up with the Ramachandran map. And he wrote this paper in 1963 along with his colleagues Shashi and Ramakrishnan, called the stereochemistry of polypeptide chain conformations. The famous Ramachandran plot is what I show you here on the left. But this is what the plot looked like when Shashi first presented it in a symposium in Madras in 1962. And then over time, the map changed slightly and eventually ended up like this. This is the evolution of the Ramachandran plot in these years. And what was being plotted in these two, on the two axes? What was being plotted in the two axes was a parameter called the dihedral angle. And I can illustrate what the dihedral angle really is, provided you come here and hold the mic in front of me while I remove my pen from my pocket. If I have two atoms, one pen will tell me the parameter that defines the structure, a bond distance. What's the distance between the two atoms? If I have three atoms, as in the structure of water, I have two distances and an angle. But 3 points make a plane, so these are all planar. Atom can be anywhere. And now instead of a plane, the structures have gone into 3 dimensions. This is stereochemistry. So what happens is that for a connected set of atoms, the dihedral angle becomes a very convenient parameter to now introduce 3-dimensionality. And it is 3-dimensionality which was introduced into a 2-dimensional plot. So the Ramachandran, analytically, you have reduced it to a 2-dimensional problem. Because all of us can understand a graph which is in 2 dimensions. We have a little bit more difficulty in 3 dimensions. We will have impossible difficulties in 4 dimensions. But how do you calculate these? Now you know how to calculate the distance. There's a formula to calculate the distance between 2 atoms or 2 points, x1, y1, z1, x2, y2, z2. There's a formula to calculate the angle. Similarly, there's a formula to calculate the dihedral angle. But these calculations become more complicated as the number of coordinates increase. All the calculations for producing the Ramachandran plot were done on this calculator over here. There were no computers. And much of it was also done with Clark's logarithm tables. And you can imagine how these calculations, how long these things would have taken. And Ramakrishnan did his PhD. He was my senior colleague, unfortunately no longer with us. It's his picture in 1967. And he produced this map for his PhD thesis in Madras. So someone asked me, should PhD thesis be published? Because the UGC says that you don't need a paper. And you don't need a paper if the thesis has the Ramachandran plot. And that's really one of the things that magnificent work of the 1960s. This is Ramakrishnan again in 2009, many years later, teaching how to calculate dihedral angles to students at IISc Pune. This was about 10 years before he passed away. And you can see that the calculation does require that you know a little bit of mathematics. But even more than that, it requires that if you do it by hand, it requires that you don't make mistakes. And you must then check your calculations. So you have to be absolutely meticulous. But where did the map get its name? This book appeared by Richard Dickerson in 1969. And this is the first book which came on protein structures, which made people, biochemists, understand. You can see he's called it the Ramachandran plot at this time. And that's how the name Ramachandran plot was not given by Ramachandran. It was actually given by Dickerson and Geiss in this textbook which appeared in 1969. These are the way the helices were illustrated. Today, computers allow you to illustrate these things so wonderfully well. But I will tell you an interesting sidelight about Ramachandran. In the famous Ramachandran paper in the Journal of Molecular Biology in 1963, the lot, nobody corrected it. Ramachandran never saw it. Many years later, when I was producing a special issue of Current Science in 1990, and this issue was being presented to Ramachandran, I asked my colleague Ramakrishnan, can we, when we reproduce this, can we correct the legends? He said, no, don't do that. Professor will be angry. So he was a sort of very 55 years to have made a typographical mistake in, we don't know who made the typographical mistake. It could have been made in the press when the paper was being printed. So the Ramachandran map allows you to study protein structures by dividing space, conformational space, and asking which areas are allowed spaces. In the morning, for the teachers, I showed you Pasteur's quadrant. So the idea of a space is always important because you need to know how to navigate space, whether it's conformational space or academic space or innovation space or whatever space you want to think about. These are the structures that you find in textbooks. All of them are understood with Ramachandran angles because they will all appear at specific positions on the Ramachandran map. Today, proteins look like this. Wonderful colored pictures. The secondary structure shown so nicely. You'll find these in books. I won't spend time on them. But in order to understand them, you use the Ramachandran angles, the phi and psi angles along the polypeptide backbone. Proteins are complicated because they are long molecules. This is a photograph of David Phillips demonstrating the structure of the first enzyme stalled, lysozyme, the third protein structure ever, at the Royal Institution in London in 1965. And you can see he's got a model here where he stretched it out to the ceiling, and then he's got the correct model determined by X-ray diffraction to show you compaction in biological structures. Compaction requires molecules to undergo contortions. Today, these are the assessments internationally. Rose 2011 says no general biochemistry textbook is complete with the phi-psi map without the phi-psi map of the alanine dipeptide. This iconic plot is a compact representation of a profound organizing principle, one that ranks among where people try to modify the map. They say that we find no need to invoke additional interactions to explain the backbone conformations of proteins. The van der Waals interaction was enough. Now it turns out that there have been occasionally comments on this. This one by David Eisenberg said that Pauling and Corey probably understood this map, but they felt that there was no need to explain them with a map of this sort. There's no evidence for this. But if you look, sometime later, Jack Dunnett asked the question whether Pauling really understood the configuration of amino acids and the effect of the configuration or chirality of amino acids on the handedness of the helix. Everything in nature is chiral, one-handedness. This you might think about because the newspapers are having articles on this possibility. Discussions have actually started. But it turns out that Pauling very cleverly, he knew what it was, so he didn't draw the alpha helix for an optically active amino acid, he drew it for glycine, which is optically inactive, the only one. So handedness did not matter. Today the Ramachandran map is used as a guide to check crystal structure coordinates. This is, for example, what wrong structures would look like. This is what right structures look like. So it's a validation tool now. After 50 years, you can see that there are many, many views of the Ramachandran map now. And lastly, I'm coming to the end. If you take the famous papers which came out in this area, beginning with the 1963 paper, you can see the citations which have been done. This has been carried out for me by a scientometrics expert at Jindal University near Delhi. And you can see that in the 21st century, the number of citations is growing very dramatically. You can also do a Garfield historiograph, which will place these papers right at the center of a developing field, which means that these papers were seminal in the development of structural biology. In 2009, when the Indian Institute of Science celebrated its centenary, we were fortunate enough to be able to produce a postage stamp. We produced two of them. One of them we put the building, and the other one we wanted to put the picture of Jamshed Tata. The postal department then told me that you can't put the picture of a single individual. So I asked, he said, government has issued a circular, you can't do that. So I asked them, can you put the picture of more than one individual? They said, yes, there's no circular which says you can't put the picture of more than one individual. So we decided to put many individuals. And then we chose people who were alumni or associated with the history of the institute in one way or the other. Some of them are highly recognizable figures, some of them are completely unrecognizable. That I won't tell you who they are, simply because you can make this into a subject of a quiz competition for other than Raman. So there was Raman right there, and then there was Ramachandran. And then you can see I have smuggled in the triple helix of collagen into a postage stamp. He started the department in which I spent my entire career. Here's a picture of the department just before I joined it, just after Ramachandran had started it. That's what it looked like. And there were not many people. This is a picture that was taken just a few days ago, 2024, when the department assembled for its annual seminar, and they had asked me to come and talk about research done over the last 40 years. And you can see we have become more colorful, we have become more numerous in number, and we are looking more modern. But here I have put what the department looked like in 2016. And in 2016, I put the one colleague who is missing in this picture now. That again is a very distinguished son of Kerala, Professor M. Vijayan, my long-term colleague. I knew him from day one. He had also come to this meeting, although he was by that time incapacitated to some extent, and was always the life and soul of the department. If there were some things that Ramachandran would have been very happy about, it would be that Ramachandran had this idea that in addition to the theoretical contributions that were made in Madras, when he came to Bangalore, the department must make a lot of experimental contributions. And Vijayan, of course, realized the dream of introducing protein crystallography to India, and today there is protein crystallography all over. So imagine what it looked like when he first started it. I believe he would have smiled as he is doing in this photograph. This is a very rare photograph, one in which he's smiling broadly. And with that, I will conclude only acknowledging the two institutions which have provided me shelter for my entire professional career. The Indian Institute of Science in the top left, and in retirement, the National Center for Biological Sciences at the bottom right. Thank you very much.
Thank you, Professor Balaram, for such a thought-provoking and inspiring lecture and taking us back to the history of invention. Now the session is open for the discussions.
Sir, thanks again for a wonderful lecture. With the complete elucidation of the infective protein of the coronavirus, can we think about a better vaccine? Because there are a lot of allegations or doubts about the vaccines that were in the market to manage the situation during the COVID time.
Are you asking me if the vaccines work?
Indirectly.
Indirectly. I suspect they don't work as well as everybody would like to imagine that they work. They probably work to some extent. But some vaccines, I think, require a lot of post-mortem now to see. We did not collect enough data. We didn't organize it. And to some extent, in the COVID-19 pandemic, like everything else, politics came first and common sense came second. And so, for example, if you take an inactivated virus vaccine, which is what Covaxin is, if you take an inactivated virus vaccine, you take the virus, you can't now inject a live virus into a person, so you have to inactivate it. You inactivate it by treating it with a chemical. The chemical that you treat it with is beta-propiolactone, which modifies all the lysine residues on the surface of the protein, and maybe other residues too. But when you do this has been established. It's been established for the Russian Sputnik vaccine, which uses the same procedure. And this was known previously for the RSV virus. So the result of this is one has to ask the question, how effective was the chemically inactivated viral vaccine? So the same kind of questions could be asked about the others. People who were infected with the coronavirus showed no symptoms at all. And which is why asymptomatic infection spread during the pandemic. I like to think of the pandemic as an experiment in biological evolution, in viral evolution, which has been carried out by nature on an animal model. The animal model are the human beings. And it is the only example that
So I have another question related to the same thing. Like generally when we look at proteins, because the animal itself or the organism itself will die if it doesn't protect that. So in your case, I can see the protein ranging from smaller one to gone up to that level. So how does that, maintain that, or how does the virus, make
See, all viruses which are enveloped viruses which use this huge conformational transition, this is actually, there's a metaphor used for it. This mechanism is called the spring-loaded mechanism. So it's like a spring which is, what happens for example, in the coronavirus itself, is that if you take viral sequences of all coronaviruses, not the one which did the pandemic, everything, those which infect animals, all the viruses which are known, is much more conserved than the receptor binding mechanism. Because see, the protein must infect, and in different hosts it infects different things. So that's variable. Whereas the fusion, a biophysical process, is more or less the same mechanism that's conserved. See, Darwinian conservation or Darwinian natural selection works at the level of molecules, not only organisms.
So, can I again add to this? So, do you think the binding efficiency is decreasing its virulence, or it's in terms of spike protein?
Binding affinities to receptor will be advantageous to the virus, but not beyond a point. Because what is most important to fall off, there has to be a proteolytic cleavage before virus infects. All the discussion which you have about whether the virus is an engineered virus or a natural virus is based on this one proteolytic site called the furin cleavage site. That's the only thing. There is an unusual furin cleavage site. The question is, was it deliberately introduced or was it introduced by accident in evolution?
Sir, how artificial intelligence will influence the protein structure prediction?
See, for example, this year's Nobel Prize, which has been given, is for the work done at Google DeepMind on producing this program called AlphaFold, which predicts the three-dimensional structure of proteins. Dramatic success of artificial intelligence. And while specialists might have some problems with triumph of structural biology, because structural biology is one field in which a huge amount of data was generated, and this huge amount of data was organized, curated, and stored and made publicly available. Similarly, molecular biology, because huge amount of sequence information, because of DNA sequencing, was made available, stored, curated, and made available. Artificial intelligence cannot work in the absence of good data. And this field provided the data. The other things that you see of artificial intelligence now, ChatGPT kind of things, that is really by taking information from the internet, good or bad, and correct or incorrect. But if there's a very large amount of information, I think algorithms like, say, protein structures have provided the first example of the potential of AlphaFold of AI-based methods. There'll be others.
But why do you think it is not happening now? That kind of, kind of research or that kind of within India, why it is not happening? Because the legacy, what you showed is amazing. Madras being the center point for protein crystallography, protein work. But that is not happening at present at par with Indian scenario or Indian science. Why do you think that is happening?
What can I say? If one compares the history with the only history that we really look at, which is political history, these two will have influences on one another. You can see that the decline of universities everywhere, institutions in general, has happened since the late 1960s and early 1970s. And that's the period in which the Indian political system also underwent major changes. And that kind of turbulence will reflect on every aspect that one has. So maybe once again it will come when things stabilize after this period of turbulence. Yeah.
So you had chosen this peptide structure, molecular biology and all. So was it your passion or that you came into this field in some way?
If anyone asks me such a question, I'll give the honest answer is this. When I got a job, in those days, you were interested in getting a job. I'm sure that's still true. So I was happy when I got a job, because that means you'll get a monthly salary. And the kind of designation that I was given tells me what I'm supposed to do. I was called a lecturer, which means I have to lecture. I have to learn how to lecture. No one should do research. Yeah? And in Ramachandran's department, everybody was doing research. So you'll also do research. Then the question is, what do you do research on? I realized quite early that if I were going to do research effectively, and I was trained in chemistry. So I had to spend some time understanding what they're doing. But once I did that, then I must tailor my work such that it is of interest to the other people. So eventually I ended up collaborating with almost everybody in the. Don't say that these things are organized, no.
Sir, for future generations like us who are interested in structure biology or protein structure and all.
And you learn this if you have given advice to government and all the time your advice is thrown into the waste paper basket. Okay. Then giving advice to, one is government, the other one are young students. Because young students also have the mind of their own. And I think they should just do what they want to do and not be guided by necessarily by peer pressure of one kind or the other.
Sir, in your perspective, how will be the future scientific world will be?
What will science look like in the future?
Yes, scientific world.
If you have lived as long as I have, you will know that never answer such a question. You know why? 50 years ago, I've imagined Google, cell phone. Today you have information here. We never knew that we didn't even have a landline. We didn't have any telephone. We wouldn't have imagined all of this. So I don't think in science you can actually imagine what's going to happen and what would be the advance. So I suspect you should only be in a position that when an advance happens, you should be able to use it effectively.
And life.
How did he influence my career? Well, he certainly influenced my career by giving me a job. And I shall also tell you how he gave me a job, because he'd actually advertised a position. And someone in India cut that advertisement out and sent it to me by post. So I applied. And but the advertisement was not meant for me. It was meant for one of his own students who had done some wonderful work with him, and he was working with him in Chicago. And but he didn't come. He didn't want to come back from the US. And so, since he didn't come, they gave me the job. And that's the first way he influenced me. The second way, once I came here, I found that I've never seen a person like him. He was interested in only one thing, and that was science. He was completely focused on science. He will be very difficult person, very temperamental, get angry very quickly. You can't be a student in his class and close your eyes. You can't not submit the assignment. He would get angry. And even with the faculty, he always had to be worried that he might explode. But if there was a scientific result, he would listen to it for any length of time, and he would show a lot of interest. So that showed me that there are people whose real driving interest is only science. He wasn't looking for a job. He didn't want to become something else. No, he was only interested in that. That's what made him different from the other people.
Thank you all for a fruitful discussion. Thank you, Balaram sir, for your scientific explanations. CSTE to hand over a memento as a token of love and appreciation to our esteemed guest, Professor P. Balaram.
remarkable afternoon, we invite Dr. P. Harinarayanan, Senior Principal Scientist, KSC STE, to deliver the vote of thanks. Over to you.
President of the function, Professor Joby K. Jose, distinguished guest, Professor P. Balaram, Member Secretary of KSCST, Dr. A. Sabu, Sri Professor Anup Kumar Keshavan, Professor Suraj M. Bashir, dignitaries of the dais, my dear friends. Indeed, it was a great tribute to the last 1 hour. Actually, Dr. Balaram was paying tribute by talking to us or lecturing to us about the work done by, real work done by Professor G.N. Ramachandran. In a effective manner, even I am not a biologist or a chemist, but I understood what he has told. In a simple, very simple manner, the last 1 hour, he has given us an immense lecture, a legendary lecture, a legendary tribute to G.N. Ramachandran. So be with us, the KSCST, the Kannur University and the Coordination Committee of the Kerala Science Congress would keep on record our gratitude to being with us. Professor Joby K. Jose, the Kannur University Registrar, was here with us. He has delivered the presidential address. I thank on behalf of Kannur University and behalf of the organizers for giving the presidential address. Of course, Dr. A. Sabu is the General Convener of the 37th Kerala Science Congress and also the Member Secretary of KSCST. Was the person behind bringing Professor Balaram here and making this lecture a fertile one. I thank Professor A. Sabu for giving the introduction to Professor G.N. Ramachandran. So thank you, sir. Of course, Professor Anup Kumar Keshavan has introduced the speaker. Now you know who is Dr. Balaram now very well. By his way of introduction, and more than that, how Dr. Balaram itself has introduced us G.N. Ramachandran. I thank Professor Anup Kumar Keshavan for interval. I thank on behalf of the organizers and KSCST for organizing this function and also delivering the welcome address. Thank you, sir. I should thank all the faculty members, the members of the syndicate, the students here, other persons from the KSCST, officials from other departments. I thank each and everybody else who have been here for the last 1 or 2 hours for listening to Dr. Balaram and making this event a grand success and paying tribute to Professor G.N. Ramachandran. I thank the media, the Kannur.
Thank you, Dr. P. Harinarayanan, for the thoughtful words of gratitude. Before we officially conclude today's program, we would like to take a moment to express our heartfelt gratitude to all the dignitaries, participants, organizers, and media who had made this event possible and truly memorable. Now we invite everyone to gather for a group photo to commemorate this special occasion. Let us come together to capture this memorable moment.